Hello, my name is Shaina Caplan, and I am a third-year undergraduate in the Trio McNair Scholars Program and the Aoki Coastal Ecology Lab, which is housed within the Institute of Ecology and Evolution at the University of Oregon. Here, I'm presenting my poster for the 2026 Undergraduate Research Symposium. For my research project, I've been studying seagrass wasting disease and productivity in eelgrass meadows of the Coos Estuary. The seagrass Zostra marina, or eelgrass, is an intertidal and subtidal species of seagrass. We care about seagrass because it provides essential ecosystem services to coastal environments, including nutrient cycling, primary productivity, and habitat provision for marine life, including economically important organisms like shellfish. Eelgrass is imperiled by seagrass wasting disease, which is caused by the pathogenic protist Labyrinthula zoster. Seagrass wasting disease also inhibits photosynthesis in eelgrass because it consumes chloroplasts, causing necrotic lesions on eelgrass blades, which I've shown in Figure 1. Ultimately, seagrass wasting disease can kill eelgrass shoots, leading to meadow decline. It is important to study how seagrass wasting disease affects eelgrass because research shows that climate change conditions, like warmer water temperatures, can contribute to increased seagrass wasting disease. A previous study conducted in Washington State found that seagrass wasting disease leads to reduced growth rates and rhizome non-structural carbon concentrations. Rhizomes, which are shown in Figure 2, are below-ground modified stem-like structures that store non-structural carbon, and this non-structural carbon is used for the plant's energy instead of its physical structure. So, for this study, we wanted to look at the relationship between seagrass wasting disease and eelgrass productivity, which we measured through blade growth and rhizome non-structural carbon concentrations. Based on previous research, we predicted that shoots with seagrass wasting disease would exhibit reduced growth and rhizome non-structural carbon concentrations. This study is important because there is a lack of research on the effects of seagrass wasting disease on the blade and shoot level. And it gives us important information about how the Coos Estuary eelgrass meadows compare to eelgrass meadows elsewhere. To conduct this experiment, we set up 30-meter transects at 3 locations in the southern Oregon coast's Coos Estuary: Barview, Fossil Point, and Velino Island. Figure 3 shows a map of our 3 sampling sites within the South Slough, which is an inlet of the Coos Estuary. At each location, we took note of site characteristics like shoot density, which is shoots per meter squared, and percent cover, which is the amount of seagrass cover along our transects. At Fossil Point and Barview, we marked shoots for growth using the needle punch method, which involves inserting a needle through the lower part of the outer sheath of the eelgrass shoot, which allowed us to later measure blade growth beneath the needle scar. Also, we collected shoots with intact rhizomes at all 3 sites. These were the same shoots we marked for growth at Fossil Point and Barview. In the lab, we measured the youngest 3 blades for growth and visually assessed the blades for disease lesions. We then scanned diseased blades and later quantified disease severity with the AI application ELISA. Here, disease severity is the percentage of diseased blade area. We also measured the area of each rhizome, ground the rhizomes into a fine powder, and used chemistry techniques like the hot ethanol extraction method and a phenol sulfuric acid assay to extract and quantify the rhizome sugars. Moving on to the results, we can see in Figure 4 that disease severity showed no relationship with rhizome sugar concentrations, which we expressed as a percentage of total sugar content in the rhizome sample, in weight of sugar to dry weight of rhizome sample. On the x-axis here, disease severity is on a logarithmic scale. Also, in Figure 5, we can see that blade growth was also not correlated with rhizome sugar concentrations. Blade growth was measured as specific productivity, which is the percentage of daily new blade area, and is expressed as a logarithmic scale in this graph. Also, Figure 6 shows older diseased blades grew significantly less than younger non-diseased blades. In this figure, blade rank refers to the age of the blade, with first being the youngest and third being the oldest. On the y-axis, specific productivity is on a logarithmic scale. And finally, Figures 7 and 8 demonstrate slight differences in sugar concentrations and disease severity. In Figure 7, we can see that there were significant differences in rhizome sugar concentrations between Barview and Volino Island, and also between Fossil Point and Barview. Figure 8 demonstrates that there were significant differences in disease severity between Fossil Point and Barview. Again, disease severity is on a logarithmic scale in Figure 8. Characterizing site differences this way is important because it provides context to the local environmental variability that may influence our results. To conclude, contrary to our hypotheses, neither disease severity nor blade growth showed a significant correlation with rhizome non-structural carbon concentrations. Also, the eelgrass we sampled in the Coos Estuary appears less diseased than the eelgrass meadows sampled in Washington, where the previous wasting disease productivity study was conducted. The Coos meadows may be less diseased because of factors like high shoot density, high light availability, and cooler water temperatures, since all of these factors have been associated with reduced seagrass wasting disease. Higher shoot density may be associated with reduced disease severity because disease shoots often die, leaving less dense, more diseased meadows. Also, cooler Oregon water temperatures can inhibit the growth of L. zosterae and kill it. Colder water temperatures can also facilitate the development of phenolic compounds, which are secondary defense metabolites that play a role in eelgrass's immune response against wasting disease. Lastly, higher light availability leads to increased photosynthesis and growth, and may inhibit the growth of Labenthula zosterae. Also, we sampled in early summer, meaning that high photosynthetic activity could have increased the amount of sugar accumulation in rhizomes relative to blade growth. In other words, sampling in different seasons may have produced a stronger relationship between blade growth and rhizome sugar concentrations. However, the relatively healthy eelgrass meadows in the Coos Estuary could be used as seeding sources for restoration elsewhere, and again could provide important context to the spatial and temporal variability in eelgrass responses to wasting disease. And while our study was limited to one location in one growing season, repeating this experiment elsewhere is important to validate our results. I've included a couple QR codes here for my references in the ACE Lab website. And I'd like to thank everybody who has supported my research, including my mentor, Dr. Lillian Aoki, and my fellow lab members, Kate Kelly, Rachel Dennis, and Christine Burnaby. I'd also like to thank the McNair Scholars Program, including our program director, Dr. Bell, and our program coordinator, Denise Elder. I'd also like to thank my fellow McNair Scholars for supporting my research, along with the College of Arts and Sciences Experiential Learning Opportunities Scholarship, and South Slough National Estuary Research Reserve for allowing me to sample between their long-term monitoring transects at Foligno Island. Thank you.